This week we'll be taking a look at Chapter 11, which we will learn about how cells reproduce, or a process called mitosis, also called meiosis if we're talking sperm and egg making. The beginning of our chapter has an interesting story about a woman named Henrietta Lacks, who died of cancer at a very young age of 31. She went in and had been diagnosed with ovarian cancer, and some doctors had saved her cells, and 50 years later her cells are still alive and reproducing almost out of control in laboratories around the world. Most human cells grown in laboratories would die within a few weeks, but these cells seem to proliferate endlessly and have been used to study cancer. They've been used to study even the polio virus. There's a picture of Henrietta Lacks with her husband right there, and that's her son, who found out years later that his mother's cells are still being used to this day. Here's an interesting quote. Biology is the only subject in which multiplication is the same thing as cell division. So when we talk about cells, we often say they divide, which in that case, it does mean that they multiply from going to one cell into two. Why do cells divide? Well, they need to reproduce. When one cell duplicates itself from one cell to two cells, that's called asexual reproduction. Some organisms who are only one cell to begin with, that's the sole way that they're able to reproduce. Within ourselves, we have cells that are reproducing all the time, such as our skin cells every 30 to 40 days. Our stomach cells every 2 weeks. It's almost like we get a new stomach lining. They reproduce to grow so that the organism can get bigger, so that a zygote can grow into an individual, and then they reproduce for repair and renewal to fix what has died or been damaged. Cells reproduce by dividing. Division of a eukaryotic cell typically occurs in a couple steps. The nuclear division, which is when the nucleus divides, followed by the division of the cytoplasm. This sequence is called the cell cycle. One cell cycle has a few different phases to it. It includes interphase, which is the longest, mitosis, and then cytoplasmic cell division. Mitosis is the part of the division where the nucleus divides, and then followed by the cytoplasm. I've included this picture below to remind us that a cell actually isn't just a circle with a bunch of fluid in it. There's many other things inside that cell, including this extensive network of protein fibers called microtubules. And these are all part of the cell division, in which a cell needs to pull itself apart literally from one cell into two. And these proteins assist that. Most of the cell cycle is in interphase, when a cell is growing and just doing what it normally does as a cell, its normal job. But it is growing. It may be making more cellular organelles within the cytoplasm. and it also needs to replicate or duplicate its DNA to prepare for division. Interphase consists of 3 stages: G1, S, and G2. The G stands for gap, so this is actually gap 1 phase. S stands for synthesis, and then G2, or the gap 2 stage. In fact, about 90% of a cell's cycle is in interphase. And here's a neat diagram to visualize what that looks like. G1, the gap 1 stage, synthesis, and then gap 2, and then just a very small part of cell division, the mitosis, and cytokinesis, or division of the cytoplasm, or this part called the mitotic phase. G1 is the first interval, or the gap 1, the time before DNA replication, when cells engage in their regular metabolic businesses. Synthesis is the time when DNA is replicated. After all, after a cell divides, each cell, also called daughter cells, are going to need the DNA instructions, so DNA must be synthesized and copied. In the gap 2, or the second interval, the cell will make more proteins that will drive cell division and continue to grow to get ready for cell division. The nucleus divides during mitosis, which produces identical copies of its set of chromosomes. You'll remember this picture to remind us what chromosomes actually look like. This is a duplicated chromosome ready to divide. Each side of this is a copy of each other, also called sister chromatids. So this will separate at the centromere right here when mitosis occurs. Remember that human diploid cells have 46 chromosomes total, or 23 pair. You get one of each of those from your mom, one from your dad. Except for the X and the Y in males, each of the pair are called homologous chromosomes. And again, you'll remember this picture from earlier. This is a set of female chromosomes with their homologous partners together. 2 number 1s, because you get one from mom and a one from dad, and so on and so forth. 2 X's, again, means female. Here's a male karyotype showing the homologous partners here. The 1s, the 2s, all the way down to the X and the Y, who are the exception to the rule. They are not homologous. The X chromosome has thousands of genes, while the Y only has about 30. When a cytoplasm divides, the new nuclei produced by mitosis are packaged then into separate cells called daughter cells, and should have a full complement of the instructions within its DNA in the chromosomes. And the whole process will start over again in G1 of interphase. Mitosis and cytoplasmic division increases the body size during your development time, when you are developing in the uterus, after you are born, while you're growing into adolescence, and so on and so forth, and continues to replace damaged cells that need replacing, or dead cells. Many species of plants, animals, fungi, and protists reproduce by asexual reproduction using mitosis and cytoplasmic division. Remember that asexual reproduction is the mode by which offspring arise from just a single parent. Again, the cell cycle starts when a new cell forms by division of the parent cell, and then ends when that cell completes its own division. The typical cell cycle proceeds through intervals of interphase, mitosis, and then cytoplasmic division. During interphase, a chromosome is loosened to allow transcription and DNA replication, which we talked about last week. Remember that usually chromosomes are tightly packed coils of DNA around histone proteins, but they loosen in order to have transcription and replication completed. When mitosis begins, duplicated DNA packs tightly together into chromosome shapes. Here's a duplicated chromosome down here. Then mitosis itself is divided into a number of different smaller phases, including prophase. This is a stage of mitosis when chromosomes condense. This is a great time when we can start seeing them under a microscope. And then they become attached to a newly forming spindle. The spindle is part of those protein microtubules I showed you earlier. Those microtubules start to migrate in kind of a spider web shape to help move the chromosomes into place to where they need to be in the cell during mitosis. Then there's metaphase. That's the stage when chromosomes are actually aligned midway between the poles of the spindle, so basically into the middle of the cell. This is important because we want to separate these chromosome pairs, these sister chromosomes, from each other into the separate cells. Then there's anaphase, where those sister chromatids are actually pulled apart to the separate or opposite poles. And then telophase, when those chromosomes arrive at the spindle poles and then decondense, and the nucleus actually reforms in the cell. Most animal cells have a centrosome, a region with 2 centrioles that get duplicated just for prophase. Basically, these objects are more of those protein microtubules, but in more of quite an interesting shape that I'll show you in a few pictures from now. And these help to direct cell division. During prophase, one of the 2 centrosomes moves to the opposite side of the cell, and the microtubules extend from both centrosomes to form a spindle. Again, the spindle is dynamically assembled and disassembled network of microtubules that moves chromosomes during nuclear division. Here are some pictures. When you gather a bunch of microtubules together like this, they make the centrioles. And here's 2 of them. The whole area together with the centrioles in here, the centriole pair, is called the centrosome. Over here, we can actually see a cell who's ready to divide, and we can see the chromosomes. They've condensed. And then these little fibers here are the microtubules, or the spindle, that are going to help pull these chromosomes apart. In prophase, the DNA again starts to condense. The centrosome gets duplicated. That's early prophase. In later prophase, the duplicated chromosomes are visible. We can actually see those with the microscope. Even better if we stain them. One of the 2 centrosomes moves to the opposite side of the nucleus, and the nuclear envelope breaks up. In metaphase, the nuclear envelope is no longer there, and chromosomes are at their most condensed, most visible. This is probably one of the most recognizable stages of cell division. The spindle microtubules assemble and bind to chromosomes at the centromere, and the sister chromatids are attached to the opposite spindle poles. And later metaphase, we'll see our chromosomes become aligned right in the middle of the cell, just like this in the picture. This is called the metaphasal plate right here in the middle, and we can see each sister chromatid is on either side of the metaphasal plate. Our spindle fibers are ready to go, attached to the chromosomes at the centromere, and our centrioles have done a good job of directing this and are now at each opposite pole. And pretty soon, these sister chromatids will be pulled apart. Anaphase, when the spindle microtubules separate the sister chromatids, moving them to the opposite poles, and each sister chromatid would now be an individual chromosome, unduplicated. In telophase, chromosomes will reach the poles and they'll decondense, meaning they'll unwind from those histone proteins. At that time, they'll be called chromatin. A nuclear envelope will then reform around each cluster and mitosis will end. And there's a little review of what we've just talked about. A cell's cytoplasm will divide after mitosis to form the two cells, and that's called cytokinesis. Here's a little bit what that will look like. A contractile ring of actin and myosin filaments. These are protein filaments that actually form the same proteins in our muscles. They're little contractile proteins. And they form at the midsection here, forming this cleavage furrow, and it'll pinch the cell in two, going from one original parent cell to two daughter cells. Then the spindle will disassemble, and a ring of filaments attached to the plasma membrane contracts. Here's some neat pictures. I kind of remember cell division with this statement here, IPMAT, for interphase, prophase, metaphase, anaphase, and telophase. And if we were looking at a cell under a microscope, this might be what we would see. In interphase, our DNA is not within chromosomes, it's just inside the nucleus here. It's called chromatin at this time. There's our centrosomes here with the centrioles inside. We can still see our nucleus. The cell is basically just doing its normal metabolic work and growing, of course. Then in this picture we have the spindle starting to form, and our DNA has wrapped around those histone proteins and made chromosomes, and we can see them. They've condensed. That's prophase. In metaphase, we see those chromosomes, there's the sister chromatids, being pulled to the center of the cell, called the metaphasal plate, and spindles forming really nicely, and the nucleus is breaking down. By the time we get to the end of metaphase, there is no nucleus. And then we go to anaphase. Our sister chromatids get pulled apart by the spindle. The little proteins connecting to the centromere actually walk along the other spindle fibers till they get to the other side, and we get the pinching off, the cleavage furrow here, as the cytoplasm divides for cytokinesis, and our nucleus starts to reform, our chromosomes are starting to decondense, and that is telophase here. Here's some actual pictures of cells where we can see these things happening. We actually are able to see in prophase the chromosomes appear and then how they line up in the middle, and then they get pulled apart, and then the cell divides. In plant cells, it looks a little bit different because they have stiff cell walls instead of just the flexible cell membranes. Microtubules will guide vesicles from the Golgi bodies and the cell surface to a plane of division where they'll form a cell plate. And our next slide has a picture of that. This is what the cell plate looks like. So it looks like basically this plate just forms here. Rather than pinching the cell apart, a plate forms to separate these two sides. And the cell plate then will mature into two new cell walls. And there's a review of what we just talked about. Here is the root of an onion where you have a lot of growth going on, therefore a lot of cell division. And you can see all kinds of different phases going on here. Obviously, all this cell division needs control so that it doesn't run rampant. In Henrietta Lacks, the beginning of our chapter is a good example of when cell division runs rampant and uncontrolled. So there's built-in checkpoints that allow problems to be corrected before the cell cycle advances. And these checkpoints basically are checkpoint genes and protein signals that monitor whether a cell's DNA has been copied completely or if it's damaged. And if the problem remains uncorrected, checkpoint gene products can actually cause a cell to self-destruct or die. That's called apoptosis when that happens. There's a few checkpoints built in. It's kind of like a stop and go chemical signal at some critical points. Probably one of the most important is the G1 checkpoint here. Another important one, the G2. And then the M checkpoint. There'll be a go signal then if key cellular processes are completed correctly. Basically at the G1 checkpoint, also called the restriction checkpoint, basically it's whether or not it's worthy that DNA synthesis should begin. At the G2 or M checkpoint, it's whether or not the DNA synthesis was completed correctly. And if you get the go signal there, then we're going to have a commitment to mitosis. At the spindle checkpoint, we're going to take a look, are all the chromosomes attached to the spindle? Because when we divide the cell, we want to make sure that each cell gets the proper amount of DNA or the proper amount of chromosomes. And we want to make sure those chromatids separate correctly. On rare occasions, controls over cell division are lost, and division occurs over and over again with no resting period. And the cell's descendants will form what's called a neoplasm, which is an accumulation of those cells that have lost control over how they grow and they divide. Basically, they're an accumulation of abnormally dividing cells. Again, that's a neoplasm. That means our checkpoints failed. Cells of most neoplasms carry mutations, some things they have in common, resulting in an overabundance of epidermal growth factor, which stimulates a cell to enter mitosis. A growth factor is a molecule that stimulates mitosis. Cells of a neoplasm have an abnormal overabundance of phosphorylated EGF receptor. That's another protein in the cells that the neoplasms all have in common. And mitosis is being continually stimulated, just uncontrolled. When control is lost, then mutations that alter products of checkpoint genes are associated with an increased risk of tumor formation. And once a tumor-causing mutation has occurred, the mutated gene is called an oncogene. So basically, these checkpoint genes get mutated, and they don't work correctly anymore, and they don't stop the cell from continually dividing. Well, there's no control over that. Checkpoint genes encoding proteins that promote mitosis are called proto-oncogenes because mutations can turn them into oncogenes. Growth factors can create cancers, like proto-oncogenes normally activate cell division, and growth factor genes and can become oncogenes or cancer causing when mutated, and basically switched on. They're never turned off. So always, always keeping the cell dividing. Nothing to turn it off. Tumor suppressor genes normally would inhibit cell division, and if they're mutated, they can be switched off. So there's nothing to inhibit the cell from continually dividing. A good important tumor suppressor gene is the p53 gene. It seems that many cancers have that in common, that the p53 gene, that's just a name we've given that gene, is mutated. The cell then becomes cancerous and then is called a transformed cell. Again, a tumor is a neoplasm that forms a lump. An oncogene is a gene that has the potential to transform a normal cell into a tumor cell. And a proto-oncogene is a gene that can become an oncogene. Checkpoint gene products that inhibit mitosis are called tumor suppressors because tumors form when they're missing. And some examples are the BRCA1 or BRCA1 and the BRCA2 genes that regulate expression of DNA repair enzymes. Benign and malignant neoplasms, the ones that are not dangerous, not cancerous, are called benign, and those are things like ordinary skin moles. A malignant neoplasm is one that gets progressively worse and is dangerous to health and cancerous. Cancer occurs when abnormally dividing cells of a malignant neoplasm disrupt body tissues physically and metabolically. The characteristics of malignant cells: they grow and divide abnormally, out of control, and the populations may reach extremely high densities within the cell division, occurring very rapidly. Cytoplasm and plasma membranes are altered, and the balance of metabolism is often shifted. The altered or missing proteins impair membrane function. Metastasis is the process in which the malignant cells then migrate and establish neoplasms elsewhere in the body. Again, things like UV radiation, chemical exposure, radiation exposure, heat, cigarette smoke pollution, age, genetics, those are things that can cause these problems within cell division. There's 3 types of skin cancers. Basal cell carcinoma is the most common type. Those are slow-growing raised lumps that can be uncolored, reddish brown, or black. Another type is squamous cell carcinoma, the second most common form of skin cancer. It's a pink growth and it's firm to the touch and grows under the surface of the skin. And then melanoma, which does spread the fastest. These cells form dark, encrusted lumps that can itch or bleed easily. Cancer causes 15 to 20% of all human deaths. Neoplasms that are detected early can often be removed by chemotherapy or surgery before the metastasis occurs. Lifestyle choices like not smoking and avoiding exposure of unprotected skin to sunlight can reduce one's risk of acquiring these mutations that cause cancer. Remember, the cell growth, cell cycle gone awry. Built-in mechanisms monitor and control the timing and the rate of cell division. On rare occasions, the surveillance of mechanisms fail and cell division becomes uncontrollable. And tumor formation and cancer are the outcomes. So with tumors, which is a mass of abnormal growing cells, the benign tumor, remember those are the ones that are not considered cancerous, remain at the original lump site. P53, that gene, has halted cell divisions, and most do not cause serious problems, and if needed can be removed by surgery. Malignant tumors, though, leave their original site, meaning the cancer can spread and can lose attachment to nearby cells, can be carried by the blood or by the lymph system to other tissues, where they'll invade and start growing and make more tumors, and that's called metastasis. And they impair functions of organs then throughout the body when they invade like that. For treatments, there's high energy radiation and chemotherapy. Radiation tends to kill rapidly dividing cells, so that's what radiation targets. Chemotherapy acts then to stop DNA replication and stops mitosis and cytokinesis and stops blood vessel growth. Blood vessel growth is important in the fact that it brings nutrients to those rapidly dividing cancer cells and literally feeds them and keeps them going. But chemotherapy can help by stopping their nearby blood vessel growth. Because these treatments target cells that are growing very fast, it also targets some of our healthy cells that just naturally grow fast, like our hair cells, our stomach cells, etc. That's why people feel sick to their stomach when these treatments are being given. There are some drugs that are being developed to help, such as those that act as proteins or enzymes that are found only in cancer cells. Gleevec is an example, a treatment for adult leukemia and stomach cancer, and it's one of the first successful drugs targeting only cancer cells. Basically, Gleevec helps to block ATP. And in the end of the chapter, we are brought back to Henrietta Lacks' Immortal cells, revisited there. The overexpression of the Aurora B protein in cancer cells that correlates with shortened patient survival rates, and drugs that inhibit Aurora B are being tested as potential cancer therapies.